All right, welcome back, everyone. We're at the second and final lecture of Chapter 24. Remember, previously I had said Chapter 24 is a pretty long chapter, but we are really only interested in the basics of metabolism and carbohydrate metabolism. The rest of the chapter we will not be lecturing on, and you are not responsible for knowing for our class. So, we're going to go ahead and dive in, as always, with our not really attendance questions. How many membranes does a mitochondrion have? When one side of a membrane has more positive charges than the other side, what type of gradient is that? And adding protons, H+, to something causes it to become more what? Go ahead and try to answer those. Hopefully you paused your video and came up with some answers for those. Let's see what we have here. How many membranes does a mitochondrion have? It has two. It has an outer membrane that's relatively smooth, and it has an inner membrane that's very folded up. We said some differences about those membranes and some other details about the mitochondrion back in Chapter 3 when we were learning the organelles. When one side of a membrane has more positive charges than the other, what type of gradient is this? That's called an electrical gradient. The charges are different on each side of the membrane. And adding protons, H+, to something causes it to become more what? Causes it to become more acidic. The more H+, the more acidic something is, or the lower the pH is. So we're going to have some, just a little bit of a review here, but then we're going to do a lot of drawing. So I recommend that students draw along when we draw in this class. It helps it kind of cement in your mind. So it's a good idea to have 3 or 4 pieces of paper and some colored pencils for the next little bit. But let's do just a very, very basic review here. First, the rest of this lecture is going to be cellular respiration, which is focused on carbohydrate metabolism. Carbohydrates, which really is our primary source of energy. So carbohydrates, every carbohydrate that we eat is going to be converted to glucose at some point. So it doesn't matter which carbohydrate we eat. Once it's in our body and metabolized a little bit, it's going to be converted to glucose. That glucose will then either be used right away or put into storage, which we'll see at the end of this lecture. But we are going to be talking about glucose today. Glucose is the primary energy source for cellular respiration. Now, some other things do go through cellular respiration, but they do it just a little bit differently. But ideally, we're going to be looking at glucose because that's what our body uses as the primary energy source. And there we have the chemical equation for cellular respiration. We saw that back in Chapter 2, but now we're going to kind of learn what does it mean. By the end of this lecture, that big, long equation that kind of looked very intimidating back in Chapter 2 is going to hopefully make complete sense, and we'll be able to come back to it and look at each part and say, okay, so that's what that meant. But what we had was over on the left, that was glucose. That's why we eat, is to get glucose, to get some sort of carbohydrate. Oxygen, this is why we breathe. We said that that's actually the only reason that we breathe. Oxygen is for one single step in ATP production. Everything else in the body, oxygen can be very detrimental. But we're going to see today, why is it that we breathe oxygen? So those are the things that go into cellular respiration, glucose and oxygen. Then we have the act of cellular respiration. We go through the reaction, and as a result of the reaction, we get some water as a byproduct. We actually create water. It's not quite waste, but it is a byproduct of cellular respiration. 6 molecules of carbon dioxide, CO2, when we breathe out, and we're breathing out carbon dioxide, that's where the carbon dioxide comes from, is cellular respiration. And then we originally just said energy, but now we're going to make that a little bit more specific. We're going to say we get ATP, that's the energy that we're after, that's the energy that our body runs on, and heat. Remember, when we have some sort of conversion from one type of energy to another or one form of energy to another, it's inefficient, and some of it is lost as heat. And that is why we start to get hot when we're working out or doing something physical. Down here we have a mitochondria. We have the outer membrane, this kind of orange colored area, very smooth. We have the inner membrane, which is this yellow kind of folded back and forth. Those folds are called cristae. That's to increase surface area, by the way, as folds often are used in the body to increase the surface area to give more room for reactions to take place. There's a spot in between the outer membrane and the inner membrane called the intermembrane space. So that's this area right here, this area right here, the intermembrane space. So today we are going to be talking about the inner membrane, the outer membrane, and the intermembrane space quite a bit. Now, there is some open area inside the inner membrane. This kind of pale yellow here. That area is filled with this jelly-like substance called the matrix, or the mitochondrial matrix. Today, we will talk about the outer membrane. We will talk about the inner membrane. We will talk about the intermembrane space. We will talk about the mitochondrial matrix. And we will talk about the cytoplasm, which is the area outside of the outer membrane. All of those areas are going to be mentioned a lot in today's lecture. So make sure you understand what those terms are when you hear them. Now, we're going to draw, but before we do that, I want to talk just a moment about glucose. C6H12O6. C6H12O6, glucose. Now, glucose, remember how it looked back in chapter 2 when we learned about it? It was that kind of 6-sided ring because it is a hexose sugar. It has 6 carbons. It has 6 sides. Now, we are also going to talk about the carbons themselves. So even though we write C6H12O6, remember we mentioned in organic chemistry, one of the things that we do is just look at the carbon. They like to draw the carbons, but not a lot else. So sometimes you will just see C, 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 and that is because the carbons are what we're after. The carbons are the important part here. Now, to make this even more efficient, sometimes the book will instead draw a carbon as a black ball. So glucose in your book will look like this. Like that. So when we see something like this up here at the top, when it's showing our glucose, sometimes it will just be those 6 little black balls. So this image here is from our textbook, but we're going to draw this out so that we can actually see what it is that's going on step by step. I'm going to bring up the whiteboard here. And let's start with our glucose. Now, I like to draw glucose as 6 carbons. So that's how I'm going to draw it. And now it's important to realize that when we're drawing cellular respiration, we're going to be following what happens to this glucose, but it's going to go through several different steps. So I'm going to break it down into 4 steps. Some sources break it down into 3, some sources break it down into 4, but we're going to look at why the difference is while we draw. I'm going to break it down into 3 steps, and I'll mention where that 4th step comes in sometimes. So the first step that we're going to draw is called glycolysis. Glycolysis. Let me draw that a little bit better. And like I said, we're going to start with our molecule of glucose. We're going to draw our 6 carbons. That's glucose. I'm going to write that over here. There's our glucose that we start with. Now, right now, we are out in the cytoplasm. This whole process of glycolysis is going to take place in the cytoplasm. So let's write that up here. Everything that we draw for glycolysis is going to take place in the cytoplasm. Now, we're going to take this glucose and we're going to get energy out of it. But the first thing that we're going to do is actually take an ATP and attach an ATP to each side. We're actually going to phosphorylate each side. And as we phosphorylate each side, what happens? If you use ATP to phosphorylate something, what does ATP become? It becomes ADP. And the same thing over here, ADP. We now have a 6-carbon chain with a phosphate on each end of it. ATP, phosphorylated glucose on one side, ATP, phosphorylated glucose on the other side. We now have glucose with 2 phosphates stuck to it. And we have 2 molecules of ADP, adenosine diphosphate. What type of phosphorylation is that? Well, we moved phosphate from one molecule onto another molecule. So that is substrate level phosphorylation. Now, we do not need to know the name of that big, long 6-carbon 2-phosphate molecule. It is called fructose 1,6-bisphosphate. You do not need to know that. There are going to be a lot of terms here. I will tell you which ones you need to know, and I will highlight the ones that you need to know. But all we've done so far is we've actually spent some energy. Remember, the point of this is to make energy. We have not done that. We have spent energy. We have spent 2 molecules of ATP. Why did we do that? Because we want to break this glucose down. We've just provided it with energy to do that. So, now we've broken that middle bond. That's what we want to do. We want to start breaking this glucose down. We spent some energy in an effort to do that. We have 2 3-carbon compounds with a phosphate group on each end, but we want to make sure that those 3-carbon compounds do not kind of stick back together. So what do? Well, we're going to take some inorganic phosphate. Remember, inorganic phosphate is phosphate that's floating free in the cytoplasm. It's not stuck to anything. And we're going to attach that inorganic phosphate to the other end of that 3-carbon compound so that we now have 2 3-carbon compounds with a phosphate group on each end of it. Now those carbons are not going to form back together into a 6-carbon compound. You do not need to know the names of these compounds. They seem similar, but they are not. I'm not even going to give you the names of them. But why did we do this? Well, now it's time to get some energy. We're going to have 2 molecules of NAD plus come along. Actually, I'll just write 1 on each side so that it's clearer. Remember from last class, we talked about NAD+. What does it do every time we see it? It's going to do the same thing. It's going to come along, take some electrons. It's going to take some protons too, but electrons are what we're after. NAD+ comes along, takes some electrons from that 3-carbon 2-phosphate molecule. And when he does, what do we call him? We call him NADH plus H plus. NADH plus H plus. Once we remove those electrons and protons, we can now harvest our energy. We're going to have 2 ADP and another 2 ADP. This 2 ADP is going to come out and it's going to grab those phosphate groups. We're going to move those phosphate groups off of the 3-carbon compound onto the ADPs. What do we get? We get 2 ATPs. We've just made some energy. Same thing over here. We're going to take those phosphate groups and stick them on the ADPs so that now we have 2 ATP. And we are left with 2 3-carbon compounds. There's no risk of these sticking back together because we've removed some electrons and some protons, and now they don't really like each other too much, so we don't have to worry about them coming back together on their own. We can have removed those phosphates without an issue. These 2 3-carbon compounds are both called pyruvic acid, or sometimes you'll hear it called pyruvate. Pyruvic acid or pyruvate. And that is the end of glycolysis. That's just the first step of respiration. So let's see the important things that we want to take away from this. I will circle them in blue so that we see all of this stuff happen, but what's the really important takeaway? Well, first, step 1 is called glycolysis. Step 1 is called glycolysis, and it happens in the cytoplasm. We're not even in the mitochondria yet. We're outside of the mitochondria in the cytoplasm of the cell. We start with a molecule of glucose. Along the way, we lose 2 ADP. I'm going to write that over to the side. Negative 2 ATP. We lose 2 ATP. We lose 2 molecules of NAD+. Those NAD pluses are filled up with electrons and protons. We got 2 ADP as a byproduct. Now we take 2 ADPs on each side, so 4 ADPs total. And what do we make? 4 ATP. So we spent 2 ATPs, but we gained 4 ATPs. What does that mean? Means we have a net gain of 2 ATP. And that's the really, really important part, is we gained 2 ATP. Now, that might sound like a good deal. We made 2 ATPs during this step. We made 4, but we spent 2, so really we have a net gain of 2 ATPs. But we're going to see that 2 ATPs isn't really much. 2 ATPs wouldn't do your body any good. That's all we got so far. And at the very, very end, we are left with 2 molecules of pyruvic acid. We are now going to follow that pyruvic acid through the next couple of steps. That is the end of glycolysis. The blue stuff is what you want to take away from this. Now, we are going to follow one of those pyruvic acids. We're going to go to what I call step 2A. Step 2A, which is called the transition phase. Sometimes you will see it called the preparatory phase. But step 2A, the transition phase. And the transition phase is going to happen as we cross both mitochondrial membranes. So we're starting with our pyruvic acid. Now we're going to look at one of those pyruvic acids. But remember, at the end of glycolysis, we got 2 pyruvic acids. So for every glucose that we start with, we're going to go through the transition phase twice, once with each of the pyruvic acids that we made. So here's our pyruvic acid. Now I'm going to draw 3 things that happen. And I'm going to draw them one after the other after the other. In reality, they all 3 happen at about the same time. It is one very quick set of reactions that happen simultaneously, but I'm going to draw them one at a time. First, coenzyme A. Remember coenzyme A from our last lecture? He is a molecular chaperone. Every time we see him, he's going to do the same thing. He's going to come along and attach to a molecule and hold that molecule together as it goes through some other reactions that might cause it to fall apart. But he's going to hold that molecule together. Now, at the same time, an NAD plus is going to come along and do what he always does. He's going to take some electrons and some protons, become NADH plus H plus. And when that happens, a molecule of carbon dioxide is going to be lost as waste. At the end, we are left with a 2-carbon compound because we lost one of them as carbon dioxide. CoA, coenzyme A, attached to a 2-carbon compound. This is called acetyl CoA. Acetyl CoA. So let's see what's important on here. This is step 2A, the transition phase or the preparatory phase. It happens crossing both mitochondrial membranes. We start with a molecule of pyruvic acid times 2, because remember, this is happening to that other pyruvic acid that we made at the same time, just off screen. Now, as we cross the outer membrane and the inner membrane of the mitochondria, these next steps happen. A molecule of coenzyme A attaches itself to the molecule to hold it together, to act as a chaperone. A molecule of NAD+ comes along, strips some electrons and some protons, and becomes NADH plus H+. We lose a molecule of carbon dioxide, CO2, as waste. And as we are crossing that inner mitochondrial membrane, we are left with a molecule of acetyl CoA. Everything on this screen happens twice. So we start with 2 pyruvic acids. We gain NADH plus H+, twice. We have CO2 produced as waste, twice. We end with 2 molecules of acetyl CoA. All of that once for one pyruvic acid, all of that once for the other pyruvic acid off screen. Once we get to acetyl CoA, now we're going to enter the mitochondrial matrix and step 2B. Step 2B, called either the citric acid cycle or Krebs cycle. Citric acid or Krebs cycle. Step 2B, citric acid cycle or Krebs cycle, happens in the mitochondrial matrix. So we have now arrived inside the mitochondria. We have our molecule of acetyl CoA, and waiting at the door is a 4-carbon compound called oxaloacetic acid. Yes, that's one of the terms that you do have to know. Oxaloacetic acid. It is a 4-carbon compound waiting as acetyl CoA arrives. When that happens, coenzyme A is going to pass these 2-carbon compound off to oxaloacetic acid, and now we have a 6-carbon compound called citric acid. Citric acid. Coenzyme A leaves to go do the same thing again. But citric acid, a 6-carbon compound. Now, even though this is a 6-carbon compound, it is not glucose. Remember, we have lost some protons, some electrons, and some carbon dioxide, so oxygen, along the way. Yes, it has 6 carbons. No, it is not glucose. Once we have citric acid, which is the first thing that's produced, which is why it's called the citric acid cycle, a few things are going to happen. First, all of the carbons and the hydrogens and oxygens that we don't see are all going to switch places. It is still a 6-carbon compound. It still has the same number of carbons, the same number of oxygens, the same number of hydrogens. They all just rearrange themselves. This is called isocitric acid. You do not need to know that one. You don't need to know the name. You just need to know that citric acid, everyone in there kind of plays musical chairs. Everyone switches places. The reason we do that is so that it is easier for NAD plus to come along and leave with some electrons and protons as NADH plus H plus. When that happens, a molecule of CO2 leaves as waste. So we are left with a 5-carbon compound. This one you do not need to know the name of. It's called ketoglutaric acid or alpha ketoglutaric acid. You don't need to know that name. But what happens here is we have coenzyme A. What does he do? He always acts as a molecular chaperone. We have NAD plus come along. What does he do? He always leaves with electrons and protons. And we lose a molecule of CO2. We are left with a 4-carbon compound with coenzyme A attached. This is called succinyl-CoA. You do not need to know that. Succinyl-CoA. Now this next step, there is a lot that goes on. It is a kind of complicated process that I don't think you all need to know. If you want to know about it, you can read about it, but they actually don't teach this part until kind of some upper level college classes. So what I like to do is I say some magic happens and we gain a molecule of ATP. If you are interested in what happens there, you can text me, you can email me, and I'll let you know. But we are just going to say some magic happens and we get a molecule of ATP. Coenzyme A leaves to do his job all over again, but we are left with a 4-carbon compound. This is called succinic acid. You don't need to know that. It is just a 4-carbon compound. I drew 5, so no, I didn't. That's an arrow. So, 4-carbon compound. Here, for the first time, we're going to see our friend FADH2 come from FAD. FAD comes along, takes some electrons and some protons, leaves as FADH2. Nothing else falls off, so we are still left with a 4-carbon compound. It is a different 4-carbon compound because we lost some electrons and protons. This is called fumaric acid. You don't need to know that. Just like we saw over here with citric acid, everyone kind of switches places. Still a 4-carbon compound. All of the carbons, the hydrogens, the oxygens shifted places. An NADH comes along, leaves with some electrons and some protons as NADH plus H plus, and we are left with a molecule of oxaloacetic acid, who is now waiting for the next molecule of acetyl-CoA to come in, and we go through this process again. Everything we draw on this board has happened twice. Once for the one molecule of pyruvic acid that in the previous phase became acetyl-CoA, and once for the other molecule of pyruvic acid that became acetyl-CoA off screen. For one molecule of glucose, we go through the citric acid cycle 2 times. So what are the important things here? There's quite a bit of important stuff on this one. First, it is step 2b. The citric acid cycle happens in the matrix. I will point out now, some sources combine steps 2a and 2b. They combine transition phase and the citric acid cycle into one big thing. That's why some sources say there's 3 steps, some sources say there's 2. 3 versus 4. I break them down into 4. I break the second step into 2a and 2b. We start with a molecule of acetyl-CoA times 2. The first thing we do is produce a molecule of citric acid times 2. We have 1, 2, 3 molecules of NADH plus H plus produced times 2. We have a molecule of FADH2 produced times 2. We end with a molecule of oxaloacetic acid times 2. We have 2 molecules of CO2 produced as waste times 2. And we produced 1 molecule of ATP times 2. That ATP was produced through substrate level phosphorylation. Okay, so a lot going on this board, on this bit of the whiteboard. This is not the last step. But everywhere so far we have followed the carbon compounds. If we see here, no carbon compounds have left the cycle to go on to the next phase. Why is that? Well, even though we keep talking about the carbon does this, the carbon does this, it's not the carbon that we're after. It is the electrons that the carbon carries. On all of the steps so far, it is the NADH plus H pluses and the FADH2 that we're really concerned with because they are carrying those electrons. And those electrons are the real important players here. So let's go to the final step of cellular respiration. This step is called it's step 3, the electron transport chain, or ETC. And the electron transport chain happens along the inner mitochondrial membrane. The inner mitochondrial membrane. So this one is going to be more of an art work, a little bit more of a drawing than what the previous were, where we were following chemicals. Here we are drawing things out to see the process. So first thing I'm going to draw is a blue line that represents the inner mitochondrial membrane. Now, above the inner mitochondrial membrane is going to be the intermembrane space. Below the inner membrane is the mitochondrial matrix. Intermembrane space up top, mitochondrial matrix down below. Blue line is the inner membrane, that one that's folded up a lot. Now, along that inner mitochondrial membrane, there are a lot of proteins. Here's one protein, and he's got kind of a weird little side protein on there. Here is another protein. There's a third protein, and then there's going to be a weird-shaped protein over here at the side. That looks kind of like that. Now, the inner mitochondrial membrane, there are a lot of protons. I'm going to draw a lot. There is no significance to the number that I'm drawing, so don't try to make it match exactly the number of H pluses here. This is just meant to represent a lot. Now down here by the third protein, there is a lot of oxygen. And I'm going to write it the way that our book writes it. And that is 1/2 O2. And there's a lot of it. 1/2 O2. Now, what is 1/2 O2? Well, it's O. It's oxygen. There is a reason why our book writes it that way. When we breathe and we inhale oxygen, it is not oxygen atoms. It is molecular oxygen. It is O2. It is two oxygen atoms stuck together. Those two oxygen atoms stuck together are what we breathe. But by the time it gets to this point, they have been broken apart. They are now oxygen atoms. Our book writes 1/2 O2 just to kind of show you where it came from. It is the oxygen that we breathe, but it is no longer O2. It has been split. And lastly, I'm going to draw our NADH plus H plus and our FADH2s. All of those that we have been producing for the glucose that we saw and for the glucoses that we didn't see, all of those NADH plus H plus and FADH2s are gathering in the mitochondrial matrix. So what happens? Where does this ATP come from? Well, gathered around this weird-looking protein, there's a lot of ADP. There is a lot of inorganic phosphate, but that doesn't really mean much. What do we have to do to get them stuck together? They won't stick together on their own. Let's look to see what happens to all of this NADH plus H plus and FADH2. Well, what are the NADH plus H plus and FADH2 doing? They are carrying electrons. They're carrying protons too, but they're carrying electrons. They are electron carriers. And they are in their full state. So they have electrons in them. They really like electrons. But that first protein over on the left, he really likes electrons even more than NADH plus H plus. So any electrons that NADH plus H plus is carrying, if it gets near that first protein, he's going to steal those electrons. He's going to steal those electrons. And that weird little side protein, he also really likes electrons, but his shape doesn't allow him to take them from NADH plus H plus. He does allow them to take electrons from FADH2, and then they are passed into that first protein. So all of that NADH plus H plus and FADH2 that we produce along the way goes to the mitochondrial matrix, and then all of his electrons are stolen by that first protein. Now, I'm going to start really stressing some terms here, and it'll sound silly, but I want to make sure that everyone understands that I'm saying two different things. Those electrons are in the first protein, but remember, NADH plus H plus and FADH2, in addition to carrying electrons, they're also carrying protons. What kind of charge do they have? Positive. What are they attracted to? Negative. So those protons are attracted to the electrons in that protein. So we're going to draw H plus, because they are attracted to those electrons. Now NAD plus and FAD are empty. They can go back and do their job again. This protein, the first protein, once he has electrons, he is a kind of ejector. He holds on to those electrons and those protons. But what happens? The second protein likes electrons even more than the first. All of the electrons that move into that first protein are stolen by the second protein. The protons want to come along, but that protein is a one-way tunnel. The only way that those protons can leave are through the top, going into the intermembrane space. So follow those electrons and protons. The electrons being carried by NADH plus H plus and FADH2 move into the first protein because the first protein likes them really, really, really a lot more than NADH plus H plus and FADH2. The protons come along attracted to those electrons. The second protein likes electrons more than the first protein, so they move over. The protons, no longer being attracted to that, are kicked upwards into the intermembrane space. That's why there are so many protons in the intermembrane space. Now, if there happen to be any protons that were not able to follow the electrons fast enough into that first protein, well, they will move into the second protein when the electrons are there. The third protein likes electrons most of all. He will take any electrons that are in that second protein. And again, H pluses, it's a one-way tunnel for them. They are ejected upward into the intermembrane space. This is constantly happening. Electrons moving into the first protein, getting taken by the second protein, getting taken by the third protein. But once they're in the third protein, what happens? They're stuck there. How do we get them out? And we need to get them out. Because if we didn't get them out, the whole process would stop. There would be no electron transport chain, which is what we're looking at. We could see these electrons getting transported from one link to another link to another link until they get to that last one. How do we get them out? Why do we want to get them out? Well, down at the bottom of that third protein, we see a lot of oxygen. This is what I keep alluding to. This is why we breathe oxygen. This one step right here. What do we know about the properties of oxygen? He really likes electrons. He is very electronegative. He likes electrons more than that third protein. All those oxygens gathered down at the bottom, they're going to take those electrons. Those oxygens are going to get some negative charges. And when they have negative charges, what are they going to be attracted to? Positive charges. All of these H pluses up at the top, think what do we have going on? One, we have a chemical gradient. They want to come through because there's a lot of protons up top, not a lot of protons down at the bottom. So there is an electrical gradient. The H pluses want to come inward. They can't. Remember, all of these things are one-way paths for those H pluses. There is also an electrical gradient. A lot of positives up top, but because of these O negatives, we have a lot of negative charges in the matrix. The positives are attracted to the negatives. That is an electrochemical gradient. An electrical gradient inward and a chemical gradient inward. But we can't get in, can we? That's the reason for this last guy right here, this weird looking protein at the end. This is called ATP synthase. ATP synthase. Protons can fit through there. It's kind of like a tube, and those protons go through the tube and out the other side to get to that oxygen. And two of those H pluses will attach to every oxygen. What do we get? 2 hydrogens for every oxygen. 2 hydrogens for every oxygen. 2 hydrogens for every oxygen. H2O. There's our water byproduct. Those H pluses are coming in because they are attracted to the O minuses. But for every H plus that comes through this weird shaped protein, it causes it to turn. It turns, it rotates. And as it rotates, it kicks this little weird arm outward. And every time that weird arm kicks outward, it takes an ADP and an inorganic phosphate, and it slams them into each other hard enough that they stick, and now we have ATP. Lots of ATP. For every molecule of glucose that we start with, we get somewhere in the neighborhood of 28 to 32 ATPs. Much more than we saw before. Remember, we had 4 in glycolysis, a net gain of 2. We had 2 produced for the 2 turns of citric acid cycle. We have 28 to 32 in the electron transport chain. So, electron transport chain happens along the inner mitochondrial membrane. It is step 3. You do not need to know the numbers of NADH and FADH2. All we need to know is just that they are carrying electrons and protons. The electrons move through the electron transport chain. You want to know the details there of how that happens. You want to follow the electrons and the protons. We make a bunch of water, and we make 28 to 32 ATP in the electron transport chain. Altogether, we get somewhere in the neighborhood, and different books will give you different give you different values, but it's somewhere in the neighborhood of 38 ATP, give or take, for the entire process from glycolysis all the way through the end of the electron transport chain. So electron transport chain, 28 to 32 ATPs. The whole process of all cellular respiration, around 38 ATP, give or take. Now that was a lot going on. None of that chemistry was new, but you do need to know that chemistry. That is something that you have to know. So, let's go back. Here. This is how your book draws glycolysis. That's what we drew on the first slide. Here is how your book draws both the preparatory phase or the transition phase and the citric acid cycle. I have them side by side to show you that it happens twice. Here is how your book draws the electron transport chain. Here showing you kind of a big picture, showing you all of the steps at once, what goes in and what comes out of each one. But now let's look at what happens when maybe the whole process, something disrupts it. Well, one thing is we can start to feel burn if it happens a lot. If we go through the electron transport chain, or if we go through the whole process a lot, then sometimes we feel the burn. If you've ever worked out, you've either had or heard someone talk about feel the burn. Why is that? Why do we feel a burn sometimes when we're working out? Well, oxygen is needed for everything that we just saw. Let's go back to our drawing. Oxygen down here is the final electron acceptor. He is the guy who pulls the electrons out of the electron transport chain so that the electron transport chain can keep going. If it wasn't for oxygen, then what we would have is electrons would stay in the electron transport chain. We would not be able to pass them through because this last protein would get full of electrons. The second protein would get full of electrons. This first protein would get full of electrons. If they're all full of electrons, they can't pull electrons from NADH plus H plus and FADH2. So those get full of electrons. The electron transport chain stops, which also means we can't have the citric acid cycle because there would not be any NAD pluses to come along to make NADH plus H plus. If that happened, if the citric acid cycle stopped, the transition phase would stop. We would not have any NAD pluses to take electrons. We would not be able to make our acetyl-CoA. We would be stuck with pyruvic acid. As pyruvic acid starts to build up, then what ends up happening is some of the electrons and protons are put back on to the pyruvic acid, trying to free up some NAD pluses. When we put those back onto pyruvic acid, it becomes something called lactic acid. Glycolysis can still progress in the case of no oxygen or little oxygen. But instead, pyruvic acid is converted to lactic acid. In low or no oxygen environments, lactic acid is produced during glycolysis. Lactic acid starts to build up, and since it is an acid, and since it's building up in high amounts, it starts to burn. It feels like a burning sensation in our muscles. Incidentally, prior to a heart attack, that pain that's being felt in the heart is also lactic acid building up because there's not enough oxygen being delivered to the heart, usually due to a blockage. Sometimes it's not really a heart attack. Sometimes it's something called angina pectoris because there is some blood getting through to deliver oxygen. Sometimes it's more severe and it does lead to a heart attack. But that burn that you feel in muscles during times when you're using more oxygen than you're bringing in is because pyruvic acid is being converted to lactic acid. Lactic acid builds up and it burns. What about other things that can disrupt the whole process? So disruption of ATP production can be through metabolic poisons. Lots of different ones, but two really important, really common ones are cyanide poisoning, which if you've ever heard don't eat apple seeds, they'll kill you. Well, they do have cyanide. It would take a ridiculous amount of apple seeds to have enough cyanide to poison someone. You could eat some apple seeds and it's not going to hurt you. But if you get enough cyanide from apple seeds, or incidentally, cyanide is the poison that works in the gas chamber. So how does cyanide work? cyanide interferes with one of the protons in the electron transport chain. So let me click through here. Cyanide interferes with one of these protons, which again blocks everything so that the electron transport chain stops. Eventually it stops completely. We don't get oxygen, we don't get any movement, and that will completely stop ATP production, and that's how cyanide poisoning kills someone. There's a class of poisons called uncouplers, and the way uncouplers work is they will poke holes in that inner mitochondrial membrane. Why is that important? Well, normally when everything's working correctly, the only way for the H pluses to get through to the oxygen is through the ATP synthase, and that drives ATP production. But if we have holes, well, those H pluses can come through anywhere. There's no need for them to go through that protein at the end. So that's how they uncouple the process of H plus coming through. Now, how does our body handle glucose? Sometimes we have too much glucose than our body needs at the moment. Sometimes we don't have enough glucose coming in through our diet. So how does our body manage all that? How does it shuffle it around? Well, glucose, we mentioned one of the ways that our body stores glucose back in chapter 2 was in this long chain called glycogen. Glycogen is just a lot of glucose stuck one together after another, branching, forming long chains, long branches. That is glycogen. There's lots of it in our liver. There's lots of it in cardiac muscle, and there's lots of it in skeletal muscle. When we have more glucose than we need at the moment, it is stuck together, and stored as glycogen. That process of taking one glucose and sticking it to another glucose to make glycogen is called glycogenesis. So glycogen genesis. Glycogen is what we're making. Genesis means formation, so it is the formation of glycogen. Now, what about if it's been a while and we haven't eaten and our body needs energy? We don't immediately perish. No, we have that glycogen stored various places around our body, and if you take that glycogen and you take one glucose and cut it off, another glucose and cut it off, another glucose and cut it off, that spills out into our blood as glucose for our mitochondria to use for cellular respiration to make ATP. That process of splitting glycogen, remember lysis, to split. What are we splitting? Glycogen. Glycogenolysis. Glycogenolysis is splitting glycogen to make glucose. And lastly, sometimes we get glucose or other carbohydrates from parts of our body that are not carbohydrate. Sometimes our body will take something like fat and break off pieces of the fat that are very, very similar to glucose. Here, we are making glucose or other starches, glucose or other carbohydrates, but they are new carbohydrates. They are things that were not previously carbohydrates. So it's new glucose or new carbohydrate that we are forming. Gluconeogenesis. neogenesis. Genesis, formation. Neo, new. Glucose or carbohydrate. Formation of a carbohydrate from a non-carbohydrate source. Gluconeogenesis. And the example that we gave was taking fat, breaking it down, and creating something similar to glucose from it. Okay, I know that is always a tough lecture. So please ask questions. Watch the lecture 24, part 1 and part 2 multiple times, because that is going to be on the exam. There will be quite a few questions from this on the exam. And I know that since it contains chemistry, students freak out. They don't like it. They shut their brains off. Please don't do that. Ask those questions of me, of your tutor, whatever it takes to get this straightened out in your head. All right. One more lecture for Unit 2. We have our tissues chapter coming up next. It will be a very brief lecture because we've already covered these in lab. So everything that we talk about in the next lecture just about is going to be a review of Lab 4, so we will not go deep into any of it. I'm sorry, not Lab 4, Lab 3. All right. So take care, and I will see you next time.